I'm Christine Curcio, and thank you for listening to this tutorial today on deposit-driven AMD and overview. This is the first of 4 tutorials. Here are my supports and my disclosures. And my takeaways are that AMD is a solvable 4-dimensional puzzle visible in vivo via OCT-anchored multimodal imaging. The photoreceptors and RPE together are the sensory chip of the brain with the unique geometry of the foveal singularity. We will discuss the value of a 3-layer versus 5-layer Brooks membrane and point out the essential AMD pathology, extracellular deposits on either side of the RPE. Remarkably, drusen follow the cone vision system, and subretinal drusenoid deposit begins where rods are numerous. One hypothesis incorporating deposit lipid composition is that they both represent failed outward transport of lipids, mostly those taken up from diet to maintain the membranes of the cones and the rods. Our view of AMD has changed dramatically due to the widespread clinical use of this imaging technology, optical coherence tomography, or OCT, which uses reflections of light to achieve depth-resolved, comprehensive, non-invasive, and quick cross-sectional views like the ones on the right. Recall the Cartesian coordinate system. A fundus view in the image plane of the retina gives the dimensions of x and y. OCT adds the z dimension across the retinal layers, and eye tracking from visit to visit adds time, so it is possible to watch a slow-moving disease unfold over years. OCT provides subcellular level information, and it fills in the gaps of clinical AMD progression. For maximum utility, experimental studies must be designed accordingly with spatial precision, high resolution, and high magnification. Spatial precision will enhance the interpretation of molecular techniques and speed progress towards preventions and treatments. On the left, the OCT device with 3 micrometer axial resolution in the frame shows retinal layers most heavily impacted by AMD. This magnified view comes from another device that averages images to enhance signal and reduce noise. The many reflective bands are named by the clinical consensus terms. So are many hyporeflective bands, most currently unnamed. We believe that there is an ultrastructural basis to all of these. We can see RPE ultrastructure in a fully reconstructed RPE cell in a rapidly preserved eye. This is what we should be aware of in our subsequent studies. Today, we know AMD as a 3-dimensional multi-layer deposit-driven disease. The OCTB scan shows layers affected by AMD. The schematic shows a progression from normal aging on the left to advanced AMD on the right. The bands arise due to the precise horizontal alignment of millions of vertically compartmentalized cells. There are multiple biologically distinct layers within 100 micrometers of vertical height between the external limiting membrane, the dashed red line on the top, and the choriocapillaris endothelium below. Here is the inner segment of the photoreceptors, the outer segment, the interdigitation zone, which is made up by the outer segments and the apical processes of the RPE, the RPE layer, and the Brooks membrane choriocapillaris layers together. Now, these bands may be visible or perturbed or absent altogether in an AMD eye. Layers are added representing extracellular deposits. The SDD or subretinal drusenoid deposits are between the RPE and the photoreceptors. Basal laminar deposit between the RPE and its native basal lamina, and drusen between RPE basal lamina and the inner collagenous layer of Brooks. Here is an electron micrograph of Brooks membrane, a flat vessel wall. Brooks membrane shares commonalities with other vessel walls. It is an intima-like subendothelial space. On the right, RPE basal infoldings and choriocapillaris endothelium are shown in red. This is the two-way exchange interface between the choriocapillaris and the RPE, and from there to the photoreceptor. In this set of electron micrographs, the choriocapillaris is at the bottom and the RPE is in the top. AMD pathology is best explained if we think of Bruch's membrane as its middle three layers, the inner collagenous, outer collagenous, with the elastic layer between, rather than the traditional 5 layers. The RPE has a basal lamina with different collagens from those in the other layers. In AMD, the basal lamina thickens to become basal laminar deposit, or B-lam-D. It adheres to the RPE and opens a cleavage plane called the sub-RPE basal laminar space, where we get drusen, type 1 neovascularization, and fluid. Different monogenic disorders affect different sublayers of this complex. These fine distinctions matter because the layers have clinical consequences and they factor into differential diagnosis. This device shows 5 bands from the EZ to the RPE basal laminar Bruch's membrane. This prototype shows a thin hyporeflective band at the base of the RPE that we thought might be the RPE basal infoldings. So soon these features will be visible in living people over time. It is useful for understanding the deposits to understand the topography of photoreceptors, the rods and the cones. The photoreceptors and RPE together form a sensing chip for the brain, like what's in a smartphone. And this chip is visible in living people. Here are the cones, and here is the RPE. I invite you to peruse these wonderful chapters about how the rods and the cones, in their physiology and distribution, evolved to match the demands of the visual environment for each species. Viewed as a flat mount in the image plane of the retina, we see photoreceptors and RPE all at the same magnification. Fovea on the top has thin cone inner segments. Perifovea on the bottom has 3 times larger cone inner segments surrounded by rings of rods. The RPE has a geometrically precise cytoskeleton in both places, as well as autofluorescent lipofuscin and melanolipofuscin, 2 long-lasting residual bodies from the lysosome system. Our microscopy of human photoreceptors, shown here, is the basis of single-cell imaging in the retina. Pictures like this help launch adaptive optics-assisted imaging, like this. This work also provided complete digital 2-dimensional maps of rod and cone density, showing the rod dominance of human retina. The 6 millimeter diameter central area is affected by AMD is less than 3% of the total, and it is captured in a clinical ruler called the ETDRS grid. In the next slide, we'll look through a slice through that map. The graph shows the density of cones, rods, and RPE in color-coded subfields of the grid. Cones per square millimeter have a 5-fold gradient in the central subfield of 1 millimeter in diameter. The rods, on the other hand, go from 0 to 130,000 per square millimeter just outside the grid. The bottom panel shows a cross-section of the human fovea scaled to the grid with the vascular beds and, importantly, the yellow macular xanthophyll pigment concentrated in the foveal center and along the plexiform layers. In the central subfield, in 1993, we used these accurate methods to map photoreceptors in an age series from 27 to 90 years old in normal eyes. We found a 30% loss of rods without gaps. In AMD, we found preservation of foveal cones on top and loss of rods in the para and perifovea on the bottom. Returning to the ETDRS grid, here is the area of vulnerable rods, and this is where we test rod vision in the next slides. The sensitivity of rod photoreceptors goes down after a bright flash of light, and the speed by which sensitivity returns is rate limited by the delivery of 11-cis retinal diopsin. This is a multi-step process that involves precisely the tissues affected by AMD. In the prospective All-Star 2 study, Cynthia Owsley and I checked rod vision at 12 degrees versus 5 degrees, the two spots on the last slide. If the speed of recovery of sensitivity were equal at the two locations, they'd fall along a line of unity slope, and they do not. Rods recover more slowly at 5 degrees than at 12 degrees, in other words, close to the fovea. In these tutorials and in our work going forward, I wish to promote spatial precision by using consistent anatomical terminology. The ETDRS grid is a good start, and it conveniently corresponds to the retinal regions defined by the great neuroanatomist Stefan Polyak. As mentioned, the 6 millimeter diameter grid corresponds to the central area, or area centralis, a region of high ganglion cell density in all vertebrates. The macula lutea, or yellow spot, is 3 millimeters in diameter and has a 4:1 rod-cone ratio. So the macula lutea is rod dominant. If we study the grid rings centered on the fovea, we see the cone dominant central subfield and the highly rod dominant rod ring in the perifovea and near periphery, or near the arcades. The area of rod vulnerability and aging in AMD has a lower rod cone ratio, about 3 to 4, depending on the age, than the areas of high risk rod density. For AMD prevention and OCT relevance, let us add the element of time and find big effects in aging in the layers of AMD. In this slide, we compare findings in 20 and 90-year-old normal central retinas. On the left are the cones and the rods we just saw, minimal decline for cones and decline to 64% of youth in rods, and the flat line is the RPE. On the right is choroidal thickness and coverage of Bruch's membrane by the choriocapillaris endothelium, showing a similar change over the decades as rods. In the middle is a twofold thickening of the 3 layers Bruch's membrane and a 15-fold increase in esterified cholesterol, the main ingredient of lipoproteins in high-risk drusen. These data strongly suggest that AMD is a vascular originating disease. The photoreceptors do less well than the RPE because they are further away from the choriocapillaris. We will focus on that 15-fold increase in the drusen tutorial. I will give a preview of the 3 tutorials on AMD deposits: drusen, subretinal drusenoid deposits, or SDD, and basal laminar deposit. Drusen are a late-life manifestation of the cone vision support system, and they confer huge risk. High-risk drusen are direct precursors to type 1 macular neovascularization and atrophy, and they can be differentiated from the low-risk or pan-retinal drusen, and their localization under the macula lutea and the fovea within it is key. The risk from aging is as big as all the gene variants plus lifestyle together. One viable model is that the RPE is relatively functional and transport is impaired from the RPE, and that creates drusen. I will present data showing that RPE is capable of making lipoproteins, which may accumulate, leading to inflammation and a cellular response. There are cell culture model systems that exist to study this system, and therapeutic approaches learned from cardiometabolic disease can be explored. Subretinal drusenoid deposits start where the rods are numerous, and unlike drusen, they are risk indicators, not precursors, for type 3 neovascularization and for atrophy. They appear first near a hotspot of high rod density. They appear in older patients than those with drusen. The major component is lysolipids, possibly indicating outer segment remodeling, and they are ultrastructurally and molecularly distinctive from the cells around it and from other AMD deposits. B-LAMD was the first histologic staging system from the Sarks and Killingsworth, and the abundance is associated with progression risk. It's now visible in OCT for the first time. It's an aberrant form of RPE basal lamina material present in all AMD eyes, but not unique to AMD. It occurs in several monogenic inherited disorders. It is a prerequisite for high-risk drusen, and it traps soft drusen material in a formation called basal mounds. B-LAMD, because it can be reproduced in mouse models, provides a road to animal models relevant to drusen formation. Today we know AMD as a three-dimensional, multi-layer, deposit-driven disease. There are multiple biologically distinct layers within a short distance between the ELM and chorion capillaris, many visible in clinical OCT. Two sets of vessels, type 3 from the retina and type 1 from the choroid, each lead to exudation and atrophy, and each is preceded by extracellular deposits that also lead to atrophy. SDD starts where rods are numerous, and soft drusen material tracks the cones and their support system. There is indeed a neurodegeneration because photoreceptors and RPE do die. Here in Brooks membrane, there's an atherosclerosis-like pro-inflammatory progression. Please listen to the other tutorials for details.